Welcome back to our session focused on gene organization and chromosomes. After we have learned how DNA actually achieves this level of compaction, let us have a look why this is important. This is important not just to generate space, but this is also a nice way for the cell to regulate a level of gene expression. Please do remember what I have told you a few lectures ago. We have discussed that although all of our cells have the same genetic information, they indeed do differ. They differ in shape and they differ in function. And this is achieved by gene regulation because not all of our genes are expressed in all of the cells at the same time and at the same level. And one way of achieving this regulation is by regulating compaction of the cell. And in terms of activity, there are two types of chromatin you need to remember. The first type is the so-called euchromatin. This DNA is uncoiled and active. In contrast, heterochromatin remains condensed, close, and is active. And this actually makes sense. In order to allow transcription of a gene, DNA needs to be accessible for the RNA polymerase. Otherwise, transcription of a gene cannot take place. And indeed, those differences between euchromatin and heterochromatin are quite huge. You don't need to remember all of those molecules depicted on this slide, but keep in mind that the level of compaction in heterochromatin is much higher compared to open transcriptionally active euchromatin. So what is the difference between euchromatin and heterochromatin? First difference is the so-called acetylation. The histones in euchromatin are heavily acetylated, and this keeps the DNA open. Okay, so acetylated chromatin is open and transcriptionally active because it can be accessed by RNA polymerase, and the gene on those DNA sequences is therefore on. In contrast, in heterochromatin, which is usually deacetylated and have low level of acetylation, those nucleosomes are much closer to each other, and therefore the DNA cannot be accessed by RNA polymerase. As a result, the gene is off and the cell is not producing the gene product at this very time. That being said, heterochromatin can be turned into euchromatin and vice versa. And this is a highly dynamic process. I'm not going to be talking about chromatin modification and remodeling in detail in this course because this is not that relevant for you as a future pharmacist. But there are certain proteins that actually write the pattern of acetylation on the histones, and there are also proteins erasing them. So the system is quite dynamic. Heterochromatin can be decondensed. In contrast, euchromatin can be condensed. And this has consequences on the expression of the genes coded on those very DNA regions. Another difference between euchromatin and heterochromatin is actually methylation. Histones in euchromatin are not methylated. So this is quite opposite to the level of acetylation. Okay, so those histones are not methylated. In contrast, in heterochromatin, we have high level of methylation, and this keeps this DNA fairly closed, right? As a result, we have either low or no transcriptional activity when those DNA sequences are densely packed. So why is this important? This is important, again, because we have very, very different cell types in our body. And what you can see here is a mouse brain slice. And DNA on this slice is stained with a specific dye which stains the nucleic acids in blue. So each of those dots here represents one nucleus. We have some cells with quite large nucleus and quite faint staining, but there are also some cells with quite dense DNA, and those cells have very, very small nuclei. And the difference is basically the level of compaction of the DNA in those cells. Those cells are active. Therefore, the chromatin is open. The DNA is mainly present in those cells as euchromatin. In contrast, in those tiny cells, we have mainly heterochromatin, which is closed. So what is the difference between those cells and those cells? Those cells are nerve cells, neurons. They are transcriptionally active. They are performing their function. In contrast, those tiny, dense cells are stem cells. They are resting because in normal, healthy organism, stem cells actually don't have that much work, right? If there is normal homeostasis in a certain organ, if the organ is healthy, stem cells are resting. They are in a resting state. However, after injury or if regeneration needs to take place, those nuclei undergo dramatic change. The chromatin gets decondensed, allowing thereby the cells to express genes and perform their function to repair the organ which is affected by injury or by a disease. Importantly, DNA packaging can also be a drug target, and this is something of interest for all of you that actually think about career in pharmaceutical industry. For example, histone deacetylase. This is an enzyme which erases acetylation patterns on a DNA, has been revealed as a very, very promising drug in targeting epilepsy. Epilepsy is a result from overactivity of certain nerve cells, and if we can prevent this overactivity by condensing or keeping the DNA in acetylated states, this could actually be quite helpful. Similarly, also methylation of histones can be a drug target as well, and here it has been shown that by manipulating the level of methylation can actually prevent the proliferation of certain cancer cell types, division of those cancer cell types, and in this case the researchers have looked at pancreatic cancer cells. So this is highly relevant. In addition to regulation of gene expression, chromosomes are also quite important for cell division. They are important for the so-called cell cycle. Here, chromosomes are single long DNA molecules that contain linear array of many genes. And those chromosomes undergo very dramatic reorganization when the cells divide. As a result, single chromosomes can only be visible during cell division, during mitosis, because they are getting highly condensed, as evident on this schematic view on cell division. So in active phase, where actually cells do express genes, DNA is decondensed. And this is how DNA looks during a certain phase of the cell cycle called interphase. In the interphase, DNA is read and needs to be accessible, and this is what you can see here. So DNA is very, very decondensed and accessible for transcription. In contrast, during the metaphase or M phase, DNA is very, very condensed. So we have high level of compaction because there is actually not that much transcription going on. And this is actually the only phase of the cell cycle where we can see those chromosomes. With this, I would like to end session number 2, and I will see you again in session number 3. Thank you very much for attention.